Hello and welcome to this video on the interesting difference between inter and intra-group diversity. It is an established concept in research and observational data that populations, behavior, and even genomic traits vary more between members of the same group than between groups. This is the inter and intra-group variation. This video will help by acting as a primer to a coming video on how global population genetics vary, but that this variation is less important than you might think. Inter is used for the between-group measure, and intra is used for the difference within the same group. Taking these measures is important in research that examines both subsamples of a large population and when comparing populations. Good examples of this are the recent study of autism diagnoses and vaccines from Denmark. The researchers had to make account of the variation within a population and did so with statistical analysis tools. In particular, ANOVA and Levine's analysis. This is not to say that this concept only applies to humans. It is applicable to most everything in modern research. In research, the intra and intergroup variation performs an important role in validation. One is the degree of variability in the samples obtained from the experiment, in other experimental settings, or to the reference group in the same experiment. The other measures the variability within the same experimental group. Take gene studies as an example. They are able to pare the variables down into individual tissues and genes that each express. The variety of tissues in any body creates a wide variance in the possible distribution of genes. Think of all the different organs and cell types. Each one could have its own function or combined function, which makes it behave differently. As an example, brain tissue will have a lot of neurotransmitter genes that are active, whilst liver or muscle tissues will not have this expression. Conversely, muscle has a lot of different purposes, but it primarily uses amino acids to build protein as a major element, something the brain has far less need for. The muscles in legs are needed for strength due to continuous use, but the muscles in the pelvic floor are used less frequently. Overall, the gene expression of muscle tissues exhibits heterogeneity due in part to the common purpose, which is in stark contrast to other tissues. But the degree of activity is not as consistent. This is an overall measure of tissues. But what then of individual cells? This is where something called flow cytometry comes in, the short and simple explanation of which is that it sorts a stream of cells into their own specific category using a variety of techniques. These can then be examined by genetic function testing. For this reason, an internal reference is used for genes. This reference gene is used in most research, creates a relative expression to a chosen constant. Depending on the tissue and research topic, this can vary wildly, but the selection of reference gene is nearly always something called a housekeeping gene that has a reasonably constant degree of expression within the cell. As you can see, within tissues, depending on the purpose and situation, what they do and what genes they use can vary considerably, even within the same tissue or cell type. Now let's look at a very obvious example. On a large scale, skin color is perhaps the easiest way to see this. This is primarily driven by melanin content of the skin. If you ignore geographical spread and just use the color of every living person, you would get a relatively standardized bell curve. This would be a continuous line from very dark to very light. Intuitively, this makes sense as a spectrum of variation in people. When you start looking at local populations, things get a little strange. This creates an artificial upper and lower limit on the variation, but the population no longer creates a nice neat bell curve. Let's use Ireland as an example. You will find almost no one with dark skin there, but you will find a cluster of light-skinned people. Conversely, Central Africa will have dark skin. In each place, there will be a very different distribution of how each skin color attribute is scaled. You'll find that rather than being a nice neat bell curve, it will have an almost jagged appearance, as there will be some groups that are overrepresented and others that are significantly under or not represented at all. This oddity is one reason why people from one geographical region can pick the accent, facial characteristics, and even in some cases the gait of another, if they are not from their local area. The norm for each is far clearer for an internal member of a group than those from another group. Going from the small genomic and the larger societal differences, there is whole organism variation. Consider the appearance of the same species and even breed, such as dogs. The visually unique elements of each breed are common across the members of the breed, but demonstrate a small variation that make your dog unique compared to someone else's. That is the high degree of intragroup variation. The gene expression or behavior pattern is different enough that you can clearly identify one animal from the others as your own. Then you have the differences in breeds. There is a clear distinction between them. All German Shepherds look alike, as do all pugs, Labradors, and poodles. The between-group differences are averaged out, such that the distinctions are larger, but as a consequence, they cover fewer categories. This is a pattern seen in humans despite cultural and social constructions. To quote one study, the inter-individual intergroup discontinuity effect is the tendency for relationships between groups to be more competitive than the relationships between individuals. It has been observed robustly in studies conducted in the United States, which is a society characterized as individualistic. Another study found that intragroup and intergroup variation accounted for more than 70% of the decision-making difference between groups. On a social or cultural level, these differences are observed in behaviors. Take vegans who engage in terrorism to suppress others for political reasons. These are unlike most other populations who do not engage in such behavior. Within the vegan cult, it is likely a majority who also do not engage in such behavior, as there is a majority in the general public. This is the intragroup variation, which is relatively small. The small subsample differs drastically from the population within the vegans. You can then compare how vegan cultists behave to how normal people behave, and see that for the most part, they are similar on a large-scale comparison. Internally, each can be split and demonstrate a significant variation. Let's take this back to a simple study that you can do yourself. Consider all the people you know and those who are your friends. The people you know are likely to be a scattering of random attributes such as height, hair color, skin color, sex, and so on. There is probably no discernible pattern if you are in a multicultural society. Now look at those you consider friends. They are probably majority the same sex as you. Hair color and skin color are less significant, but there's likely to be a trend there too. As for height, you are probably all of a similar height and weight. This extends to personal interests. From here, you can see how a common set of traits may become entrenched in one particular group, and that those with other attributes will create their own nexus. This is also true in classrooms, where grades do not always follow a standard bell curve. Some classrooms may perform admirably and can get near perfect grades year-round. Others may not and struggle to even pass. This difference is a perfect example of intergroup differences. Less extreme cases are likely to show a fairly even spread of grades. This would be true in the extreme classes by the even spread, which would be skewed towards one end. When you aggregate the grades into one place, they form a nice neat bell curve. But taking individual classrooms out and plotting them against this whole data set will make clear that they break the trend in some way. This is because you begin to slice the samples into smaller and smaller parts. In doing so, the unusual or different elements become more pronounced. The converse of this is that larger groups can overshadow or blur distinctions. All of this is an interesting and odd pattern. It can explain some of the problems faced between ethnic, religious, and even cultural groups, where they have an inside and outside mentality. It is important to measure how the differences within a group function and how these compare to those differences between groups. From an evolutionary perspective, it may be a simple case of selecting for particular traits within a set population or group. The kin selection is based on a common set of features that allow the core population to retain what is considered desirable or that which provides an advantage. Take selective dog breeds, for example. Taking account of differences in a group versus between groups is an important tool for the extrapolations of findings and application of these. Lessons from one study may not always translate to another population, but on a broad strokes basis, they will generally do so if there is a degree of heterogeneity in the first group. The difference between groups is nearly always less than the difference within a group. This is important when trying to apply medical treatments or study behavior on a case-by-case basis. Such broad justifications are useless. On a population level, it is good. When looking at populations, you cannot use small samples because they cannot be extrapolated by the commonality of comparative populations. Ultimately, this is a question of granularity. How far can and should you split populations to understand different features? Thank you for watching this video. If you have found it interesting or useful, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.